In this section of the lecture, we're going to be looking at the microscopic anatomy of skeletal muscle and look at how the nervous system controls the contraction and the movement of the muscular system. We're going to start where we left off, and then we're going to be looking at smaller and smaller. So this is the gross anatomy of the muscular system. And if you remember from the last lecture, we have the epimysium, which surrounds bundles called fascicles, which are surrounded by the perimysium, which surround the muscle fibers, which are surrounded by endomysium. And so we're going to just look at a single muscle fiber. So let's say we went down to this small level here. Now we're going to pull out this one muscle fiber, and we're going to look at the microscopic anatomy of just that one muscle fiber. So this picture here is a single segment of a muscle fiber. So I don't want you to get confused, because you'll notice that inside this muscle fiber we have these little round things that look kind of similar to the picture we were looking at before. This looks like a bundle, almost looks like a bundle of muscle fibers, but instead this is a single muscle fiber, as I pulled from that other slide. And within this muscle fiber, we have these little bundles of proteins. These are the microfilaments that are going to actually be doing the contracting in the skeletal muscle. So if this whole thing here is one muscle fiber, these are the nuclei. Here's one, here's another one over here. So remember, skeletal muscle is multinucleated, and so there's a few of the nuclei. And then there's something called the sarcolemma. So because a muscle fiber is a cell, it has a plasma membrane. If you think back to the lecture where we talked about the structures of cells, all cells have a plasma membrane. Muscle cells also have a plasma membrane, but we give it a special name, and it is called sarcolemma. And if you look at the word sarco, the prefix here means flesh, or referring to muscle. So the plasma membrane of a muscle fiber is called sarcolemma. Here we have a myofibril. So within one muscle fiber, we have all these bundles of proteins called myofibrils. And we're going to pull out a myofibril and look at what makes that myofibril, the proteins that make that up. Notice here though, before we move to there, notice we have these striations. You see these lines down the length of the muscle fiber. These are the striations. So skeletal muscle is striated, cardiac muscle is also striated, smooth muscle is not. These striations are caused by the myofibrils. So when you look at a skeletal muscle, the reason you see these striations is the makeup of the myofibril. And we have what's called a dark band, or the A band, and a light band, or the I band. So the dark band is what makes that darker line, that striation, on the muscle fiber. It's not pointed out in this slide here, but there's also something called the Sarcoplasmic Reticulum. Sarcoplasmic Reticulum. And the Sarcoplasmic Reticulum, there's that sarco again. This is the endoplasmic reticulum found in a muscle fiber. So the endoplasmic reticulum in other cells has a job in producing lipids and maintaining the cell. In the case of the smooth endoplasmic reticulum, remember there's smooth endoplasmic reticulum and rough endoplasmic reticulum. The sarcoplasmic reticulum is the specialized smooth endoplasmic reticulum in the muscle cell. And it's going to come up here in a minute when we talk about nervous system control, because the sarcoplasmic reticulum plays a part in muscle contraction. So next, we're going to pull out one myofibril. So here's one muscle fiber. Now we're just going to take one piece of that muscle fiber and look at a close-up of it. And here we are. So here's our myofibril. Myo, again, referring to muscle. So this is This is a fibril within a muscle fiber. And this is what it's going to look like. I'm going to point out a few of the pieces here. Notice we have the A band and we have the I band. So in that previous slide, the A band was the dark. So a way to remember this is dark has an A in it. So the A band is the dark band. The light band of the striations is the I band. So light has an I. So we have our A band and we have our light band. Notice that the A band is made up, the reason it's dark is it's made up of these overlapping fibrils. These are microfilaments here that are overlapping. The little blue filaments and these red filaments, these are proteins. These are proteins that are produced by the cell, and they're bundled here, and they're what cause contraction to happen. We have what's called the H zone. The H zone is found in the middle of the A band. So notice if you look here really closely, and we'll look at this a little bit more in the next slide as well, but the H zone goes from where the overlap of the red fiber here and the blue protein end, and to where it picks up again. So you have this little gap here between where the blue proteins end. That's called the H zone. You also have the Z disc. The reason it's called the Z disc is it zigzags. See how it zigzags down here like this? And this is found in the middle of the I band. And then we have something called the M line. M line standing for middle. So M line is found directly in the middle of the H zone. So to review once again, we have the I band, which is the light section. Notice that the light section is only made up of these blue filaments. Then we have the A band, which is the dark section, and it's made up of an overlap of the red and the blue. H zone is where there's just red, there's no blue. M line comes right in between the H zone. And then we have the Z disc right in between. That's where the blue microfilaments hook together. Notice within this bundle here, within this myofibril, we have all these proteins all packaged together. So when this muscle cell contracts, what's going to happen is these myofibrils all at the same time are going to shorten in length. And as all these mini myofibrils shorten, and this happens within all of the myofibers within the muscle, this is going to cause the contraction of the muscle. So now we're going to go a little bit closer, and we're going to look at just one little section from about here to here. We're just going to look at one section of this myofibril. So this is that little section of the myofibril. We call this a sarcomere. There's that sarco again. What a sarcomere is, it's a section of a myofibril from one Z disc to the next Z disc. So if we go back to this previous slide, we're looking at a section from here to here, and then the next sarcomere would go from this area over to the next Z disc, which we can't see. So a myofibril is made up of all these little sections called sarcomeres, all packaged right next to each other, the length of the sarcomere. So here we have the Z disc again. The Z disc is where these blue filaments were attached. Now we're going to name these filaments. The blue ones are the thin filaments, and they are made of actin. Actin is a type of protein. So these are thin actin filaments, and then the red filaments are thick filaments, and they're made of myosin. There's myo prefix again. So the thin filaments are actin, the thick filaments are myosin. Notice here that the thick filaments have all these little projections all over the end. Looks kind of bumpy. We'll look at those a little bit more in just a minute. Notice that the Z disc holds these actin filaments in place. So they're all attached to the Z disc at the end, and then there's no attachment over here. And then the thick filaments, the myosin, here's the M line, which means that this area here is the H zone. Notice that it's attached here at the M line. If we put some other terms here, this area here is going to be the A band. Sorry. Should be A band. And then from here over to where the next A band starts, here we have the I band, the light band. So the dark band, the A band, and the I band. What's going to happen here when contraction happens, these actin filaments are going to move closer to each other. So this H zone is going to decrease in size, and that's going to cause a shortening of the sarcomere. And as the sarcomere shortens, again, this adds up over the course of the whole muscle. This is going to cause a shortening of the muscle as a whole. So each sarcomere just shortens just a tiny bit, but there's all these sarcomeres, the length of the myofibril. And so over the course of the muscle cell, this is going to cause a shortening, and so your whole muscle shortens, and that's how muscle contraction happens. Let's go to one more slide here, and we're going to look at a close-up of the myosin filaments. So you can see the thick filament, the myosin, and you see on the myosin you have all these little projections, and these are called the myosin heads. They look like little heads sticking off. The thin filament, notice it doesn't have any of these. And here we have our H zone, and our H zone does not have any little heads on the thick filament in that area. So what's going to happen when contraction happens are these little heads are going to attach to the thin filament. And it's going to work just like a little oar. They're going to move, they're going to pull, and they're going to pull these thin filaments closer together. So this is going to go this way, this is going to go this way. It's going to decrease the space between the thin filaments. That's a contraction. We'll look at that in a little bit more detail, but that's just a general overview of the microscopic anatomy of the muscle. Let's now look at how the nervous system comes into play here. So the nervous system is what controls the contraction of the muscles. So when you think in your mind, oh, I want to move my bicep, or I want to stretch my leg, your brain sends a signal down the neurons, and it triggers that contraction to happen in your muscle. It triggers the shortening of the sarcomeres that we've seen just briefly here. When we're talking about stimulation of contraction, there's some important words to learn. There's first one is excitability. So this is the ability of the muscles to receive that signal and to act on that signal. So muscles can be excited, so they're stimulated by this impulse from the nervous system. And they're able to contract. So when they receive that stimulus, when we receive that signal from the nervous system, they're able to contract. That's important. If they weren't able to contract, our muscles would not be able to move. Then also they have extensibility. They're able to be stretched. But they also have elasticity, and that's important because after the signal is stopped, they're able to go back to their resting length, their resting stage. So they're elastic. So as the nervous system signal comes, it says, okay, go ahead and contract. It contracts, it's extensible, but then when that stimulus ends, the muscle automatically goes back to its previous length. So we don't have to consciously think about uncontracting, I guess, our muscles. It's going to jump back to that previous shape automatically. I want to look briefly at how the nervous system comes in contact with the muscular system. We'll look at this in more detail in the next lecture when we talk about the nervous system but I want to give you a little overview here. So in this slide, what we're looking at, to get our bearings here, here's our bone over here, and our tendon attached to that bone, and then we have our muscle. So here's our muscle bundles, our fascicles, and here we have our muscle fibers. So each of these muscle fibers is a separate cell. Notice it's multinucleated, it's striated. You can see the dark bands and the light bands. You can see the M line in there as well. And then we have our nerves coming and associating with these muscle fibers. A motor unit is the bundle of muscle fiber and the neurons that trigger that muscle fiber. So in this case, if you look at this little red neuron, it's coming in and it's associating with this muscle fiber right here. Only this muscle fiber. Notice it's not associating with any of the others. So this red nerve and this single muscle fiber there, that would be a single motor unit. If you look at this other motor unit number 2, here, notice that it's coming in and it's associating with this muscle fiber, and this muscle fiber. So it's associating with 1, 2, 3 muscle fibers. So this motor unit is bigger than this other motor unit, but this still is one motor unit. Even though it's attaching to 3 muscle fibers, it's stimulating 3 muscle fibers, it's still one motor unit. So motor units can stimulate many muscle fibers or just a few, depending on their job. So for example, if you want to move your fingers are all moving separately. You have a lot of control with how your fingers are moving. And so there's going to be more motor units going to your fingers to have all this very minute control, as opposed to a larger muscle that simply acts all in one consort. So for example, your thigh muscle, there isn't a lot of different intricate movements that it does, and so it's going to have fewer motor units going to that area and controlling it. So depending on the movements required, you may have more or less motor units. When that axon, so a neuron has an axon. This is the axon body. So the nerve cell is up here in the spinal cord, and then its axon, which is still part of the cell, but this is just the tail, kind of. It's like the tail of a cell, and it's coming and it's connecting to this muscle fiber. I guess I didn't notice that this neuron also stimulates this muscle fiber, so it actually has 2 muscle fibers. But anyway, and so it's coming and associating here. So now we're going to look at how that associates with the muscle fiber. So here we have the axon coming down, and notice here at the end the axon can branch. So this axon is coming in contact with the muscle fiber in 3 places. So our electrical stimulus is going to come down. So our signal is being sent from the brain, and it's going to come down and it's going to attach to this muscle fiber at 3 places. That's going to signal the muscle fiber to start to contract. We call this area the axon terminal, where that axon terminates. And it terminates just above the sarcolemma. So it's hard to see here, but you have your sarcolemma and you have your axon terminal. That axon never actually touches the sarcolemma. There's this tiny space there. So that is the basic the microscopic anatomy of the muscle fibers and how the nervous system comes in and associates. So what we want to do now in the next section, we're going to look at how the nervous stimulus causes the contraction, and then we'll look at how the contraction itself happens a little bit more.